It's an honor for me to introduce today Dr. Jonkers, which is professor at the Human Movement Biomechanics Research Group at KU Leuven, which will talk us today about this problem and how multi-scale approach might actually help filling this gap and leading us to new ways for regenerative methodologies. Thank you.
Thank you very much. I want to start with, of course, acknowledging the organizers and thank them for this invitation. We have been working together, Jerome, in a couple of councils, but so far I had not the pleasure to be here. And I have to say I'm very impressed by the work of the group and the organization of the summer school. So thank you very much for giving me that opportunity. So the work that I will be presenting today is not so much my work, but it's the work of my team. It's the cartilage mechanobiology team in Leuven. And I want to take you on an adventure that started 5 years ago, approximately. And I want to show you some of our approaches trying to highlight or untangle some of the factors contributing to degenerative joint disease, and more in particular, it will be focused in many of the applications on knee osteoarthritis. I thought that I would bring you first to Leuven. I don't know if some of you have been there. This is just a picture of the town hall in Leuven. Very nice to visit. Always welcome to come and meet our group. And if you don't come for science to Leuven, then you might come for Stella Artois, because indeed we have the brewery in Leuven. So always welcome to come and sample some of this beautiful beer. But today it's not about beer, it's about science. So let's continue. So as Rami already introduced yesterday, osteoarthritis is a very common and prevalent chronic joint disease, and millions of people worldwide are affected. And it is a whole joint disease, but the damage in the cartilage is one of the hallmarks. And patients will present with pain and loss of function. And actually, if you look at the loss of years spent with good quality, it has a major impact in society. And also because of the problem of obesity, we see that there is an increasing health burden also because people are living longer. Now, at the moment, there is no cure for osteoarthritis. So once a joint is affected, there is no way to cure the joint in itself. So the current curative interventions for osteoarthritis are limited to symptom relief. People get painkillers when they have painful episodes, or in the end stage of the disease, we can use, as Rami said, a hammer, and we can just replace the joint. But of course, that is not the best solution. And being a physical therapist by background, I would like to help these people in an early stage of the disease and try to reduce the progression of the disease as soon as possible. Now, we do know that osteoarthritis is a multifactorial disease that is affected by not only mechanical factors, but also genetic factors, obesity. There are problems with gender, age. All of these are contributing to the etiology of osteoarthritis. Now, that means that at the moment we are quite convinced that a single strategy to tackle this problem will not really be the complete answer. And more we are thinking about a combination of drug-based treatment, in which we will affect the biology of the cartilage, that together with adjusted rehabilitation programs where we can tailor the mechanical loading for the joint to what the biology can sustain. And these are integrated models of care for osteoarthritis that we are trying to prepare by doing this research. So basically what we say is we need to understand how to go from joint movement and joint loading to the response of the chondrocyte that is living there in the joint. On the other way, if we know the biology of the chondrocyte, we need to relate that back to the joint movement and the joint loading, because otherwise we are not able to give effective therapy to our patient. And it is in this circle and completing this circle where multi-scale modeling has its own right to be. So I'm going to use this scheme, and it's a scheme from Andriachi. It dates back to 2014, so it's already a bit aged, but I think it's a very nice scheme in which I try to position my own research, because I want to go from joint movement to actually understanding the mechanobiology of the chondrocyte in order to understand how I can change or impact the biology, impact the tissue characteristics by affecting the movement behavior of my patient. I'm not going to say that we have completed this circle, but I'm going to show you for each of these parts some examples of the work that we are doing. Some of them involve more modeling, some are more experimentally. So let's start with the human modeling work that we are doing at the moment. So it is known in literature that the progression and the onset of knee osteoarthritis, medial knee osteoarthritis, can be related to mechanical overloading of the knee joint. In older work, a lot of the authors have focused on the external knee adduction moment, and with that we are referring to the moment that is occurring around the knee joint in the frontal plane. So we are actually evaluating how this ground reaction force factor is falling with respect to the knee joint center. And what is known is that in knee osteoarthritis, this lever arm is increased, and due to that, we say that there is higher medial compartment loading in the knee joint. So this has been described, and it is known that it is related to the progression of osteoarthritis. But if you look carefully to those studies, these are mainly studies that relate to patients who are in a further advanced stadium of the disease. These are patients typically prior to a total knee replacement surgery. We do know that they have knee osteoarthritis, they have pain. So there we are a little bit running behind the facts. We would like to have functional biomarkers for OA that we can use to screen patients and to identify the patients that are at risk for progression. Now, can we use these joint moments to identify people early on in the disease process? We did a study in Leuven in which we selected control subjects in green and also established OA patients in red. And that are the typical patients that are reported in literature. But we also recruited a group of early OA patients, indicated in blue on the graph. And these patients are patients that have minimal signs of osteoarthritis on radiographs, but they do have signs of osteoarthritis on MRI. So the structural progression in their joint is minimal. And what we see when we look at these external joint moments is that we can adequately separate between control subjects and subjects with severe osteoarthritis, but that the joint moments do not allow us to identify these early OA subjects. So in these patients in the early stages of the disease, the structural progression is not yet there, and therefore these joint mechanics cannot identify altered loading in these patients. So this brought us to the use of other modeling approaches, trying to look more in detail to the mechanical environment of the joint. So we used standard integrated motion capture, so we use camera-based system to track the displacement of different markers on the body. We also measure the ground reaction forces, and then we link this to a musculoskeletal model in order to calculate the joint kinematics, but also the joint kinetics, so these moments. But then we go one step further, and we are solving the muscle force distribution in the lower limb. So we calculate for the different muscles in the different planes what their contribution is to the movement, and from that we can then estimate what the contact forces are in the joint, as indicated by the green arrow that is pointing downwards in the joint. So from that we have a good estimate, not only from the effect of the contact with the ground, but also we can account for the muscles that are acting across the joint and that are also contributing to the joint loading. So we were very hopeful that this musculoskeletal modeling approach would give us a functional biomarker for early OA patients that we could use to identify the patients in the early disease process. But again, if you look at the difference between our green line and the blue line, so the control subjects and our early OA subjects, there was no difference. So at the whole joint level, again, we could not discriminate the mechanical environment in the joint. It was obvious, again, that in our severe osteoarthritis patients, there was higher loading, but we knew that already from our moment analysis. We were lucky to have a good collaboration with Colin Smith, who has been implementing a specific modeling approach in OpenSim, in which we can also account for the cartilage-to-cartilage contact, and in which we can use 6 degrees of freedom of the knee joint, and also account for the control of the ligaments around the joint. Using this approach and integrating that in our musculoskeletal modeling workflow, we can now have unique data on the pressure distribution on the cartilage during locomotion. So here you see how the pressure is changing during the stance phase of gait, and you can see the evolution of the pressure magnitude indicated with the colors, like in a finite element analysis, and also the location, how it changes during the stance phase of gait. So we are looking here at the contact pressure at the surface of the cartilage. We are not going into the tissue depth. For that, we need finite element modeling. Now, using this approach, we are able to discriminate the knee contact force in a medial compartment and a lateral compartment knee contact force. And indeed, what we saw was that for the first time, our early OA subjects were already different from our control subjects. So also in these people with very early signs of the disease, the medial compartment loading was increased. But something that we found even more interesting was that it was not only the magnitude of the loading that was higher, but also we could see that there was a shift in the loading pattern. So here you see the established OA patients. These are control subjects, and there was indeed a shift of the area of loading during the stance phase of gait, therefore shifting the area of contact to places that might not be predisposed to these higher loading conditions. So this means that we now have a functional biomarker for early OA. Then we thought, okay, can we use this biomarker in order to identify people that are more prone to a fast progression of the disease from those who will have a slower progression? So what we did was we took the gait assessment from a baseline condition in a group of people who had a fast progression and in a group of people who did not show any change in the Kellgren-Lorentz over 2 years. And using our medial compartment contact pressure, we could indeed indicate in this subset of patients that a higher medial compartment pressure indeed characterized the progressor cohort. And the non-progressors, they did not show any significant differences with the control subjects. So using this data from a baseline evaluation, we can identify in this group of patients already those that are at risk for a fast disease progression. These would be the patients that you might consider for a more detailed follow-up or even to put them in a preventative therapeutic exercise program. Looking at the heat maps, you can also see that in our progresses, we saw this shift of the loading location compared to the controls and the non-progressing individuals. Because there's also always this debate about, is OA about loading or is it about underloading? We thought, okay, let's look just bluntly to the difference in the mean profile of our progressors and the control subjects and the non-progressors and the control subjects. Do we see overloading? Do we see underloading? And of course, this is biology, mechanics. It's not either the one or the other. There are areas in the joint where there is a clear overloading of the joint, indicated in red, but there are also other areas where there is more an underloading of the joint. And that is interesting to try to get more insight in that discrepancy. Also, what we took from this comparison is some sort of a guideline on how much do we need to change the loading in the joint in order to make someone who is on a track to be a progressor to the track of a non-progressor? How much do we need to unload the joint? And using this data, at the moment we say a reduction of about 22% is the difference between a progressor and a non-progressor. So this for us is a first indication on how much unloading that we should try to accomplish in these patients. How to do that, that's then a different question. We are now working together with University of Eastern Finland, and we are trying to couple this to the finite element analysis in order to have a better understanding how these loading conditions that we observe at the surface of the cartilage translate more in depth in the tissue. But this is work in progress, and it's a PhD work of Ikram, who is also attending the summer school. So I heard the question this week, how can you translate this in clinics? What can we do with this? So the first example is a way of patient screening. Am I advocating that every patient has to go to a gait lab? No, we can discuss about that over a beer, but you can do other things, or wine is fine with me as well. You can do other things with this. For instance, you can start thinking about using these approaches to inform rehabilitation strategies for patients with knee OA. Because if you talk to physical therapists, they have a lot of exercises, activities of daily living, and everyone thinks he knows what the loading is in the joint. But if you start to discuss with them, no one is really sure. So what we did, we brought control subjects into the gait lab. We measured all these different activities that physical therapists are prescribing, and we looked at the magnitude of the pressure distribution and also its location in the joint. And we came up with that. Don't try to interpret this. I will make it more digestible for you. But this is a loading landscape in the knee joint for different exercises. And we were able to grade them. So this is some sort of a map that a physical therapist could use in order to determine a staged rehabilitation process for a patient. For instance, if we would consider total knee loading, this is an easy progression going from less loading to higher loading. And there are some obvious examples. I think no one is surprised that if you do a single leg jump, that the loading is higher than when you stand out from a chair. I think everyone knows that. But if we look, for instance, at the squatting exercise, that is now an exercise that is typically used later in rehabilitation. But from our data, it seems to be even loading the knee joint less than walking. So maybe this is a sign that we can introduce this earlier in the rehabilitation process. Also, for instance, if we think about compartmental knee loading, we were able to discriminate exercises that load more the medial or the lateral compartment. And this is information that now can be taken further in clinical trials in order to see if we can actually make a difference. Of course, here the challenge will be to monitor the movement execution of the patient and be sure that this balance is also safeguarded when the patient is exercising himself. At the moment, we are trying to take this even one step further using predictive simulations. With predictive simulations, you do not use any data collected from a patient, but you start from a hypothetical movement pattern in which you control the muscles in a way to achieve a specific goal. In this case, we want symmetric walking using a minimum of energy in the muscles. And we are able to get normal-looking gait patterns. But what is the nice thing about this is that we can also introduce changes in the model. For instance, we can weaken muscles and we can see how the model is adapting to maintain its walking strategy. For instance, here you see 4 different models with different levels of muscle weakness, and the model is able to find compensations that allow it still to produce symmetrical gait patterns. And the patterns that we see here in the yellow model is this typical Trendelenburg compensation that is seen in a lot of patients who have weakness around the hip girdle. So we are actually able to use the model to try to understand why people are adjusting their gait pattern to, for instance, weakness. We can also think of using this for gait retraining for knee osteoarthritis patients, because we can now start from the gait pattern of a patient indicated by the black line here, and then we can let the model find strategies to unload the joint. And we can even say, okay, now you try to stay as close as possible to the patient's own walking pattern, but still reduce the loading in the medial compartment. Or we can say, just minimize the medial knee loading. I don't care whether the patient can do it, yes or no. What would be the ideal way of walking for this patient? So we are now trying to integrate this predictive simulation approach more and more to train physical therapists in having a better strategy to affect the loading of the knee joint. So this brings me to a bit more of an exotic example. We don't only do gait assessment in humans, but also in rats. Why did we decide to go in that direction? Well, there is a big scientific debate whether it's the bone that first is affected in osteoarthritis, or whether it's the cartilage, and what the crosstalk is between changes in the mechanical characteristics of the bone and the deterioration of the cartilage. So this is a very interesting paradigm that we want to contribute by looking at this mechanical crosstalk using multi-scale adaptive models. And of course, because we also want to evaluate or validate these models, we were looking for a way of having the possibility to change the bone mechanoregulation. And of course, this brought us to the use of rodent models. But having a multi-scale model of a rat in this case is even more difficult than in humans. So in the rat, we use a surgical technique in which we destabilize the medial meniscus, and we know that 8 weeks after surgery, we have established osteoarthritis in these rats. Now, if you want to get gait kinematics of a rat, you can try to do the marker-based gait assessment, but it's very much sensitive to skin artifacts. So therefore, we have been using X-ray reconstruction. I will show you in a minute, but we will be using dynamic X-rays in order to track anatomical landmarks so that we can really assess with high accuracy the changes in the joint kinematics due to the destabilization of the meniscus. And we will use this as an input to our multi-scale modeling. So the workflow is very similar to what we are doing in humans. We have our test subjects. We use a musculoskeletal model of the rat, we try to link it to the finite element analysis, and then we will compare the mechanical local loading with the tissue adaptation that we can observe using micro-CT assessments. So here you see the setup for the gait assessments. This is our rat gait lab. We have one of the participants in our study who is feeling very happy. He gets Cheerios when he walks up and down the gate path. But what is more important is that here you see 2 X-ray sources that we use to visualize the moving skeleton. And in our walkway, we also have force plates embedded. So the data that we get from this is the following. So we have the moving X-rays, we have high definition videographs, and also here down you see the forces. So basically all the data you need for your multi-scale modeling approach. And we have been looking at the ground reaction forces in a group of rats who had sham surgery, so no intervention to the meniscus, and a group where we destabilized the meniscus. And to our own surprise, we did observe not unloading of the surgically treated knee, but there was even a slight higher vertical ground reaction force. So the animals are using the limb that has been treated. We then took the X-rays and we identified the different landmarks. This is a very tedious work, but fortunately we have machine learning that can help us out there. It's not perfect yet. We still use a lot of, involve a lot of master students there, but by combining the coordinates of the different landmarks, we can come to 3D coordinates, and these we can then use in combination with our musculoskeletal model. This is a published musculoskeletal model of a rat hind limb in which we have now accounted for the 3 degrees of motion in the knee joint, as what we would do in the human joint. So combining our experimental markers with the musculoskeletal model, we can now look at changes in the different degrees of freedom of the knee. And for instance, we can see that indeed the abduction is different in the rats that have been treated with the DMM. But what we were interested in, of course, is to come to the contact forces. So we solve our muscle force distribution, and here we already see a difference between the surgically operated rats in the dashed line and the sham surgery. But what we found very interesting is that here, in contrast to what we saw in the ground reaction forces, we found for the first time a difference and an unloading story in our rats that were treated with the destabilization of the meniscus. So looking at the medial compartment loading in our rats, we now have the impression that destabilization of the medial meniscus is not overloading the medial compartment, but in contrast, there is in our data at the moment an underloading of the medial compartment, which is interesting because everyone thinks that the destabilization will lead to overloading of the medial compartment. So we are quite anxious to take this data further into a finite element model in order to look more in detail on the local tissue strains, which we will then compare with our in vivo data. So we followed all of our rats with in vivo CT, so we can look at the bone adaptation over time. And also we did high detail micro CT with contrast agents to have a good indication of the cartilage constitution in each of these rats. So this brings me to our wish to understand the joint function, not purely from the static images, but also more looking at dynamic image acquisition, using, for instance, micro-MRI. We have been working on creating a quite unique setup in which we can compress cartilage plugs while imaging. So we bring our cartilage compression device into the micro MRI, and during compression of our plugs, we can then image and look at deformation fields in the cartilage. So this is a specific protocol that is called dense Fisc imaging, in which we look at the deformation gradients in two different planes in order to look for functional biomarkers of cartilage mechanical function. So the type of pictures you get from this is here the displacement fields. So you have the transverse displacements, you have the axial displacements, and from that we go to the documentation of the strains. And we are mainly interested in shear strains because I've been told that shear strain would be something that cartilage does not like. So I'm very much interested in shear strain here. And in particular, in this project, we were interested to see if there was a surgical debridement of a cartilage defect, whether that would introduce excessive shear strain to the cartilage tissue, and if that could be the cause of degradation in the joint. So if you look at these strain maps and you compare them with the following, and in particular with the one at your right, the bottom one, that is the shear strain at the edge of a perfectly debrided cartilage defect. So an orthopedic surgeon would try to have such a nice edge when he is doing this debridement. So this indeed shows us that in this type of procedures, these edges are really at risk of having degenerative changes. And ideally, we want now to combine this with some molecular markers that we want to take from these edges in order to further have insight in the mechanobiology of this. Of course, someone can say, well, yeah, there is a lot that can happen between intact cartilage and this macroscopic damage. If you talk about osteoarthritis, there are other things happening. You have collagen degradation, the proteoglycans are degrading. How does that compare? And can we also have an idea on how the mechanics are changing there? And can we do it through MRI, hoping that one day we can also do it in the whole joint of our patients, that we actually can look at the mechanical function of the joint rather than just looking at the structure and the geometry. So we have done this, and here you see, don't look too much at this in detail. What I want you to see is that these gray bars, these are the increase in the strains when we have collagen degradation and proteoglycan depletion in the cartilage, when we do enzymatic degradation. And really, if you have the combination of the collagen and proteoglycan depletion, that is where the tissue will fail mechanically. And we can detect it from our MRIs. The condition where we only have the collagen depletion is the one next to it. So the collagen depletion that will then trigger the proteoglycan depletion can already be characterized by increased strains in the tissue. So again, we have some functional biomarkers for altered cartilage mechanical function. We are now trying to take these approaches further to the in vivo patient knee, and we have been looking using T2 mapping and T1 row mapping and trying to correlate this to the joint loading that we see during the gait function. And indeed, there are some correlations where we were able to show that higher loading results in thicker cartilage. I don't think anyone is surprised about that, but we could also see that our higher shear loading in the joint could be related to lower proteoglycan content, whereas if there is higher compressive loading in the joint, that there is higher proteoglycan and collagen content. So with these, we try to have a better understanding of the mechanobiology at the joint level that we can then also use for the evaluation of our patients. Because indeed, finally, we would like to use this to come to some in vivo biomarkers that we, for instance, can use. This is a pneumatic compression device that we will be using in our MRI in patients, and with them, with this setup, we can then follow the mechanical function in vivo, for instance, in patients who have had ACL rupture. And combining this with the modeling approach will allow us to validate some of the insights of the models. So this brings us a bit to what can we do with modeling at the tissue level. Of course, we can use data from patients and use that to look at the mechanical environment in the joint. But recently, and also in collaboration with the group of Rami, we have been looking at using this approach to create virtual knockouts. Because degradation of cartilage is a very complex process, and there are different things going on. We see that the collagen fibers reorient. We see that the collagen fibers are degrading. Proteoglycans are affected. There is a change in the fixed charge density. But how does this complex process is happening? What is the driving component? Is not always clear, and it's very difficult in experiments to just manipulate one of these components. So we decided that we would use an in silico approach in order to selectively take out one of these degrading mechanisms and then look at the effect on the degeneration. And we did this for cartilage that was affected by osteoarthritis, so where the constitution was already changed, where we had less proteoglycans, where we had less collagen, and we did it for a focal defect in the cartilage. So we loaded both these cartilage plugs, and we looked at what happened in terms of degradation when we allowed, for instance, the collagen fibers to degrade, when we allowed the collagen fibers to reorient, but not allow the proteoglycan depletion in order to untangle the processes. And you might recognize here the first situation is primary osteoarthritis, where the cartilage is of an inferior quality. And the second one is secondary osteoarthritis, where we have a clear defect following, for instance, a trauma. And what we could differ from this analysis is that there are clearly different mechanisms playing in primary or secondary OA. In primary OA, we see that the collagen network, so the degradation of the collagen, is the driving factor that will cause all the negative cascades following from that. On the other hand, if we look at the defect model, there we see that the proteoglycan depletion is the triggering event that is causing local changes in the strain, and only afterwards there will be the changes in the collagen fibrils. So by using our models, we are able to untangle each of these mechanisms, and we hope that in a validation experiments, we are able to selectively enter on one of these mechanisms and show that we can, in our in vitro setup, change the progression of the damage. So this brings us to the last part, where we're gonna dive a bit into the cell model. And as I said, we have not closed the loop, but this is the closest we come to closing the loop. So we have been working on documenting the mechano responsiveness of the cartilage tissue and the cells in OA explants. So we have a loading setup in which we take human explants from typically from hip surgery. This is more hip related, but that's for practical reasons. And we load those explants in our bioreactor, and we have been looking at different protocols that we applied in order to then look at the gene expression in the explants. And at the moment, it seems that in our hands, a loading protocol of 10% compression that is applied for 1 hour and then followed by another session of loading is the good protocol in order to tap into the mechanical pathways. So you see, for instance, the response of the C-Jun here in gray on the top graph. Sorry, my pointer died. And also you see that we have an anabolic response in ACAN and in COL2. So we are using this to relate the loading to the molecular changes in the explant. Now what we see is when we look at the Mechano responsiveness in these OA explants, it's very nice if you look at the average value, right? You load the explant, you see upregulation of C-Jun, so we are doing something mechanically. We see upregulation of COL2, ACAN. We see that there is a change in TCF1, that's one of the genes related to Wnt signaling, and then there is also a change in MMP13 here, which we probably don't want. But if you look at the spread of the data, I think this is biology speaking, we see that there is quite a spread in each of these explants. So this triggered us to look at a correlation between the OA disease state of our explant and the molecular response. Because what we see is that if you take different donors and you look at anabolic expression and catabolic expression, that there is a very patchy pattern. So if you want to use this data for mechano-responsive regulatory algorithms, you would like to know what patient you are modeling. Are you someone who is closer to your healthy donor, or would you be willing to look at someone who is more diseased? Now, there are some patterns emerging. So the closer you are to healthy donor, the better anabolic responses you have, so that is good. And for the more diseased profiles, we see that there is less anabolic response, but what was for us very important was that we saw the hyperactivation of the Wnt signaling pathway. The Wnt pathway is implicated in the early onset of osteoarthritis. So we are very much triggered by that in order to look whether we can further detangle the role of the Wnt pathway in altered mechano-responsiveness in OA, and in particular in early OA. Because of the variability in our OA explants, we decided to do it in healthy cartilage explants and to chemically come to Wnt upregulation and then load our cartilage explants. So we are then looking at the mechano-responsiveness of Wnt upregulated cartilage. And what we were very much interested to see was that upon Wnt upregulation, the anabolic response, so we are looking on the graph to this bar, and we compare this bar is the mechanically stimulated control explant, so the healthy cartilage, and the bar next to it is the mechanically stimulated cartilage with Wnt hyperactivation. So in the presence of Wnt hyperactivation, we see that the mechanical the stimulation of the C-Jun gene is reduced. So Wnt hyperactivation seems to impede the mechanosensitive behavior of the chondrocyte. And this is then also reflected by the increased MMP13 expression in the Wnt upregulation, and also there seems to be a positive feedback loop to the TCF1 gene that shows that there is some sort of a reinforcing of the negative mechanoresponsiveness. If we then look at the anabolic genes, we see that in the normal control explants upon mechanical stimulation, there is upregulation of SOX9, COL2, and ACAN. But in our Wnt hyperactive explants, this anabolic response is decreased. There was yesterday a discussion about the pericellular matrix. Well, indeed, what we also see here by looking at Perlecan and COL6 is that the mechanical stimulation is causing less expression of COL6 and less expression of Perlecan, which are indicators of a less optimal situation of the pericellular matrix. So this is something that we want to follow in the future. Why do we want to look at that? Well, we have discovered a new toy, and that's the last thing I'm going to present. That is, we want to look at mechanics at a cellular level. So no more rats, let's go cell. So we are using at the moment traction force microscopy, because traction force microscopy allows you to look at the force between the cell and the surrounding area, so the extracellular matrix. And what you use is you use a gel-based system in which you have beads implanted, and then you will disrupt the cytoskeleton of the cell. And using those two images, the one with disrupted cytoskeleton and the intact cytoskeleton, you can look at the displacement of these beads. So actually, you are looking at the changes in the intrinsic force generation of the cell. So what we want to show is that Wnt upregulation will affect also the force generation of the cell to the extracellular matrix. We are not yet there, but we are seeing our first tractions of chondrocytes on the extracellular matrix. So here you see a chondrocyte embedded in a packed gel that is interacting with the packed gel and having force exchange with the extracellular matrix. So for us, this would allow us to come to the cellular level and look at the forces at chondrocytes. So this is where I want to conclude an overview of what we are working on in Leuven. So if you ask me, is there relevance of this multiscale analysis when you are looking at degenerative joint disease? I hope that I've been able to show you that these model-based analyses have an added value and that you can use them to look at altered joint and tissue loading, and these have applications immediately for physiotherapy and orthopedics. Also, I think by combining experimental and model-based insights, looking, for instance, at cartilage tissue strain under loading, I think that is important as a functional biomarker of mechanical function of cartilage. And also there, I see great use of these techniques for orthopedics and tissue engineering. On the other hand, looking at chondrocyte mechano responsiveness is very important because I see there the future relation to exercise programs, because a lot of these biology or biological modulators are now coming into clinical trials. So we are affecting the chondrocyte biology, and we need to know how this is going to affect our rehabilitation and exercise strategies. So for me, I think I could not live without in silico approaches. I measure a lot at different scales. I didn't put the TFM in here. And for me, using the model approach is a way of synthesizing all my experimental data and a way of structuring all that data. And based on this, I can then try to address some clinical questions for surgeons, for physical therapists, or I try to do some basic science. So I once had a discussion with a radiologist, and he said, why would you measure? Why would you model if you can measure it? And that, for me, was, yeah, you're right. But then I went back, and since then, I know I want to model because I can model the unmeasurable, and I can predict the unpredictable. And I think that now the big challenge is to bring all these model-based insights as an additional tool for medicine, and convince medical doctors, physical therapists, to also try to learn from these insights. And that is why I think initiatives like the VPH, but also at KU Leuven, we have our own Institute for Physics-Based Modeling for in silico health. That is really where preparing for in silico trials of the future, that is really where we now need to start pushing and gradually introducing this into the medical community. As I said, this work is teamwork. I'm very fortunate to have very cooperative collaborators, so I really appreciate all the research we can do together. I appreciate my team members, of course, the funding sources, and you for listening. Thank you for your attention.
It's working? Okay. Thank you, Professor. Any question from the audience? Up there, down here? Many questions, very good.
First of all, thank you so much for this presentation. Actually, I have a couple of questions, but maybe I'm not gonna go all of through them in one time. So in the beginning, for determining the least loading pattern or the least loading gait pattern and the exercises or stuff that we can maybe suggest to clinicians, was the site of the damaged cartilage taken into account, or was it the average load all out the cartilage?
That exercise program was done in control subjects. So there was no structural damage introduced in the model. Indeed, if you have a structural defect, that will of course alter the load distribution. Yeah, so, but there you also need to account for movement compensations. So that's the next step where we are now doing this in a cohort of osteoarthritis subjects. Yeah.
And for the rats, was also the muscle activity modeled as well, or was it only based on motion detection?
We have
like kinematics.
Yeah, so it's not an EMG-driven approach. It's already complex enough to make a rat walk from one side to the other. Fortunately, there is some EMG data available from literature, and we have been comparing that. But Judith is here also in the audience, so you can discuss that with her in detail. So we do look at reference activity, but it's not EMG-driven. No.
Okay, thanks. I might give.
Thank you for the presentation. I was wondering how you validate the inverse simulation musculoskeletal models when you do have some measurable data, and then my question is more about then compared to how do you measure, how do you validate the forward simulations when you are just doing the modeling?
Yeah, validation is always a big hurdle. I think it really depends what you claim that you want to do. So for the inverse analysis, we have, like everyone, used the Grand Challenge data. Yeah. If I write that in a grant, they say, you cannot use that to validate it because that's an implant and you're looking at a native joint. Point taken, yeah?
It's the best.
That's true, but it's the best I can do, okay? So that's what we've been doing, and we are there in the normal range of what we see in literature. Also, for this specific model, there was a validation done with dynamic MRI, because the ligament balance is, of course, quite sensitive. So there were passive, there was a passive movement introduced in the model, and that was compared with the images of dynamic MRI. So we do know that the ligament balancing works. And for me, that's an added value of this model, because as soon as you start tweaking geometry, you will introduce some imbalances which will affect the model outcome as well. And that's then a question, do you introduce personalized geometry? We haven't done, we are working towards that, yeah, but then comes again the validation. And then for the predictive simulation, that's hard. you cannot go in and take a muscle out of a patient and then see how he walks. We do have some quite unique data on ankle-foot movement, where we have a cadaveric simulator, where we can just pull on a muscle and then see how the foot complex changes. And we do have the kinematics documented and the pressure. So we're going to try to do that for the ankle-foot complex. But if you want to do that for a whole lower limb, I wouldn't know how to do that. But I think it all comes down to what question do you want to answer? And are you really claiming that you want to have personalized conclusions, or whether you want to have some sort more of a guideline? At the moment, I think I'm confident to say that for guidelines, you can use these musculoskeletal models. For personalized medicine, I would be more careful for now.
Amazing. Thank you so much.
Hi, I was wondering about the medial underloading in the rats. Do you think there could be some kind of a gait adaptation strategy that it happens to spare the medial side?
Yeah, we are also a bit puzzled about that. And that brings us also to the Emg pattern, because, yeah, we don't know. It's difficult to ask a rat what he's doing. We know that the ground reaction forces are the same. There are subtle changes in the kinematics, and the joint moments are different. So the fact that the muscle balance comes out different in the static optimization makes sense. What we want to do is, of course, now first look, if you put this through a finite element model, whether at the tissue level this is confirmed, yes or no. Because in this rat there is a destabilization of the medial meniscus, and indeed that will cause changes in the tissue strain. So that is now the next step. But it seems that at a joint level, they are not overloading the joint. It might be compensation, but we were, well, not surprised, because this is what I wanted to show, so I'm glad that this comes out. But yeah, if this is confirmed at the tissue level, that would be, yeah, it would change a little bit what they're doing with destabilization of the meniscus.
Okay, I'll check it out then when you guys finish. Thank you.
It's Judith's work as well.
Hi, thank you so much for your very insightful presentation. There was one study in which you said, like, we concluded that there is change of magnitude of, you can say, stresses and strains, and also the shift of loading area. So do you think this shift of loading area in OA is related to biological factors, which is happening because of OA, or?
My guess, but this is just hypothetical, is that it has to do with the ligament balance and more laxity in the joint. So we did some sensitivity studies imposing changes in geometry and changes in gait pattern and evaluating the strains in the ligaments. And if you go towards a more OA gait-like OA-like gait pattern, then you will see that the strain in the ligaments is higher, and it's also reported clinically that in many of these patients there is instability of the knee joint. So that is also consistent with that pivoting that you see of the pattern of the pressure location. So the best way of doing that is to have a good estimate of the ligament parameters. We are using now MRI, but ideally you would measure that perioperatively and then look at that. But yeah, we haven't done that yet. Yeah.
I have a question. So first, congratulations, very impressive work in general. I have a question about the MRI, that the protocol that you use to map the displacements and the strains. I was involved in a project years ago about the meniscus, and we did something similar, but with a sustained load. So basically, first we acquired the image without load, and then we compressed the leg and acquired again the meniscus, and we use image registration in order to calculate the displacements. My feeling is that you are not using image registration, right? You have a cyclic load and then you have some sort of gating to calculate the displacement, or I'm wrong?
It's an acquisition, so there is a gradient that is applied that allows you to track the displacement while imaging. So it's not a comparison between loaded and unloaded. There is a cyclic loading that is being applied, and it's through the gradient that is applied that you can actually follow the displacement field within the tissue. So it's actually an MRI acquisition protocol that stems from cardiac imaging, and it's gated. Yeah.
Okay, thank you. Great.
Thank you very much, Ilse, for this really great talk and for showing all this highly translational research. I have several questions. My first question is curiosity about the Wind pathways you're exploring. It seems that you're only exploring the non-canonical Wind pathways related with cell polarity. This is what I'm...
the canonical, eh? Are you saying non-canonical?
Well, there are three ones. There's canonical, the non-canonical related to cytoskeleton, which seems that is the one that you're further exploring through force traction microscopy. And then there is the non-canonical related to ion channels, which is probably also related with TRPV4, etc. So have you a very specific choice among these three pathways?
No, these are baby steps, Jerome. I'm a physical therapist and I'm now exploring molecular biology. The interest of Wind comes because the group that I collaborate with is our specialist in Wind signaling. And I think the relation with the cytoskeleton is the first thing that we want to focus on, also with the traction force microscopy, because I also want to see how the cell adjusts to being in that altered micro environment and see whether there is a role of altered force sensation by the cell in the impaired, in the impaired mechanoadaptation in early OA. So that is where it comes from. So that there is not a true selection, but we focus mainly now on the link with the cytoskeleton.
Okay, thanks. And the second question I have is curiosity. So when you're exploring the explants, so are you making a specific choice in terms of location of these explants on the knee joint? So a bit linking with Fabio's question. So some explants nearby the meniscus or just underneath the meniscus might be very different from explants in the middle of the joint.
Yeah, these are hip explants. So the reason for that is very pragmatic. If you take the waist after or before or during total knee replacement, there's not much cartilage left. So if you really want to have good cartilage, a hip gives you a better source for the cartilage. And also what I call the normal cartilage explants comes from traumatic hip fracture. So there we have a good source of normal cartilage. We don't have that for the knee. So a lot, and I need to disclose it maybe better. So what I show in the bioreactor is not knee related, it's hip. So and there you asked about the location. To be very honest, we take what we can get. Because you're working with the material you get. We try to standardize it and we indicate what area we take. And by doing this sort of disease state quantification, we hope some studies do it by macroscopic evaluation, where they try to evaluate the damage. But we think that a molecular profiling is more relevant there. So we try to account for that by documenting the site where we take the biopsy, and then also have a molecular profile of the cell pellets that come from there.
Thanks. Well, since we have time, I dare to snake a third question. It's about, so when you've presented when you have presented the musculoskeletal body scale musculoskeletal model, and you've mentioned then the possibility to use the information to re-educate gait. I'm wondering what are the true needs in terms of modeling, because every patient will have a different proprioception. So the strategy physical therapists will have to employ to re-educate gait might also require real-time monitoring and adjustment. Is that feasible at all?
At the moment, there are systems that do real-time feedback underlying having some sort of a surrogate model that builds on the musculoskeletal modeling approach. But these are the very high-end systems, so that is still not something that can be done in every physical therapist's practice. But I think if you are willing to kind of give in on the quality of the estimation of the loading, for instance, in Stanford, they are now working very hard on an app that you could run on your mobile phone, where you could just do a video of your patient, and then with machine learning, you can get an estimate of the abduction moment. So at that level, there are already things in the pipeline, and I think that is not gonna well, that will come out quite soon. And then the question is, how do you provide feedback? Is it just by showing the video? Do you need to do that real time? It's also something that we are looking at, for instance, with accelerometers and giving some sensory feedback to the patients. So that's an active field of research, but it's not yet for tomorrow, I think.
Thanks. Yeah, I think there is no...
Thank you for your talk. And this wonderful work. And I am curious about the lower level, in the chondrocyte level, that you are studying the Wnt signaling pathway, and you then studied the response at the CFOS gene. Mm-hmm. And I am wondering, why do you select this gene? Because it's also involved in another signaling pathways, not only in the Wnt signaling. Mm-hmm. And how do you make sure that the activation of CFOS is just come from this Wnt signaling?
Yeah, I think CFOS was together with C-June. That's just to look at whether or not we are tapping in the mechanosensitive pathways. We have a panel of different Wnt-related genes. I can show you later more in detail. So I think CFOS was more to show that we have a we tap in the mechanosensitive pathways. Yeah, that's not, indeed, it's not Wnt specific.
Yeah.
Yeah.
And how do you make sure that the Wnt is activated with mechano-loadings? I didn't...
The Wnt is activated by applying CHR. So it's a chemical compound that we add to the explants, and then the activation of one of the genes, the TCF1, is one that we use to check the Wnt activation. But there are different ones that there's a whole panel that we use for that.
Yeah, because when I studied the mechanotransaction, the Wnt was not the first approach studied in the mechanoregulation. And I was curious about, so thanks.
Yeah, the reason that we go for Wnt, there are some drugs at the moment in clinical trials that specifically target the Wnt pathway, and that's why we also consider that. Yeah.
Okay, thanks.
I just have one more technical curiosity. If I'm not mistaken, you quantified proteoglycans for the cartilage?
I want to, sorry?
You quantify, or like you measured proteoglycans for the cartilages. I was just wondering if you also quantified the glycosaminoglycans and how you quantified them.
There are different staining techniques that are being used for that, but for the details, I yeah, we can discuss over coffee.
Okay.
Yeah.
Yeah, please.
So histologically and also then through microscopy and quantification. Yeah.
So more qualitative approach for rather than quantitative.
Yes. Yes. Indeed.
Thank you.
Yeah. Yeah.
Like and like from the color intensity, you can just like quantitatively measure it. Yeah.
With calibration, yeah, it's semi-quantitative. Yeah.
Thank you.
So thank you, Ilse. Actually, we know each other, at least in silico, because we are dealing with in silico world project. So as in silico pioneer, I just two general thoughts that I would like to share with you. And the first one, in your impressive and beautiful work, you used still data coming from animal models. So my thought is, do you think that before or after we will be able to totally replace animal models? I mean, should we be able to measure all what we need to make a good in silico modeling, relying completely on machine-provided measures, without having dealt with the animal models in general? And the second general thought I'd like to share with you, and of course I'd like to have your opinion, have you tried and are you thinking about to submit for regulatory staff this in silico modeling to see if you can get some qualification advice from regulatory authorities? Because you know that if we would like to penetrate medical field, we still need to convince and to have good credibility assessment for obtaining qualification from regulatory authorities. Thank you.
That's two very relevant questions. The first one, can we replace animal experiments? I don't think that we can replace them completely, but I think with the in silico models, we will be able to do a clever design of our experiments, and that will definitely contribute to refinement and reduction. So I can see that in the future, probably you would test some conditions in an in silico model, and that that will allow you to kind of do a sensitivity analysis of the animal experiment that you have in mind. So I don't think we can be that ambitious that we can say that we can completely abandon animal experiments, but definitely a reduction. And then with respect to regulatory affairs, yeah, the reason why I wanted to start this IC Health Institute is because I really want to bring them to clinics, the modeling, but law and yeah, that's not my cup of tea. So if other people want to deal with that, I'm very glad that they want to do. Unfortunately, there are some lawyers who find that very interesting. So I think that is kind of the added value that I see of bringing all that expertise together as we do in silico world. Indeed, we need to bring it to the clinics, and then you need to go through the clinical path. I'm a scientist, pur sang, so I'm interested in the validation, but how then to bring that to legal authorities, then I'd rather give it to someone else to deal with that. So yes, we need to go through that, and I think all the development that's happening there is really very useful, and for us scientists really needed, but it's not my personal interest to kind of follow that. So I think they're having good bodies that assist scientists in that process. I think that is already very valuable.
Yes, I agree completely with you, and think about also to the cultural resistance that we have in when dealing with biomedical science. So, thank you.
Thank you.
Any more questions?
Yeah. Was there another question there? Okay. Yeah, so maybe last question, and so I just wanted to comment on what Francesco said and your answer. I think that the fact that the VPH Institute is backing up this kind of events. So tomorrow you will see we will have several representatives also of the Avicena Alliance, which is the corporate branch of the VPH Institutes. They are all non-profit organizations, but there are people so making a lot of efforts to try to integrate science, technology, transfer thereof with all the regulatory aspects. So many more things that happens at the policy level, at the regulatory level, and that's really very important. And I think it's a great particularity of this society, and I think this is really unique and wonderful. Having said that, my question is: so you told yesterday, and you also told it a bit today that you come from the field of motion analysis, physical rehabilitation, and now you've shown actually advanced computational modeling, and you're going now deep into the biology, down to the regulation pathways, gene expression. So that's a great venture, and here we have a lot of early stage researchers, and I was wondering whether you could comment a little bit on your track.
On my track,
yeah.
I can state it's a bit atypical. Maybe I didn't consider my career path well enough when I started, but that's ages ago. I think everything is always possible as long as you believe in collaboration. And that's really what I think. Don't be scared to go for your interest. You probably will not understand it, but I think everyone in this room is probably intellectual enough to make progress. So it will cause frustrations once you go outside of your field, but you will also discover many new things, and that is what gets me excited about science. So, and that's also what is, for me, the fingerprint of a true scientist. That is that you go out of your comfort zone, find people with whom you can collaborate, and then make progress. And who knows where you end up? I don't know.
Thank you, Ilse. I think it was great words to go to coffee break.